So, okay. Our speaker tonight is Dr. Stuart Weiss, and his talk is entitled Resurrection Ecology of the Xerces Blue Butterfly. The Xerces blue butterfly, Glaucospsyche xerces, occupied sand dunes in western San Francisco, but urban development drove it into extinction by the 1940s. Starting in the 1990s, many acres of sand dune habitat has been restored in the Presidio, including the presumptive hostplant of the Xerces blue, deerweed. In 2000, the year 2000, Bob Pyle published an article about resurrecting the Xerces blue using silvery blues from nearby populations. Stuart will tell the story of how a community of scientists and enthusiasts executed the introduction of silvery blue butterflies, including habitat assessments, genetic studies, selection of appropriate source populations, translocation, and subsequent monitoring. Um, let's see. I think I messed that up just a little bit. Let me go back here. So, oh, I somehow deleted his bio with the fuss with the Zoom bombing. So, Stuart, you're with Creekside Science, and I am afraid you're going to have to give us a few more details. But I do before that, I want to thank Bob for suggesting you as a speaker. So, thank you so much, Bob.
Okay. Yeah. So I was at Stanford University for about 20 years in various positions in Paul Ehrlich's lab. Then starting in 1999, I struck out on my own, or I was fledged from the nest. And I learned how to make a go of it as an independent scientist, um, and started up formally Creekside Science in around 2006. And I have 5 employees, and we do a lot of really cool ecological work, a lot of hands-on uh, restoration of endangered plant and butterfly populations. But then I also do a lot of pretty broad-ranging conservation planning for the 10 Bay Area counties. Big part of my work recently has been looking at monarch butterfly overwintering habitat, looking at the structure of the forest using some pretty high-tech tools, LiDAR and hemispherical photography. So we can now actually more design the habitat as opposed to leaving it to the chances of forest dynamics. And I'm going to talk about the Xerces Blue project here, or the Silvery Blue project. So I'm going to share my screen.
Okay, thank you so much for being with us tonight, Stu. I'm sorry
Yeah, it's a pleasure. I love talking about this stuff.
we're so happy to have you.
Okay, let me get this full screen. There we go. You should be seeing a Xerces blue butterfly there.
Yes, we are. It's perfect. Thank you.
Okay. So yeah, the resurrection ecology. Let me just so a little bit of the setting here. The Presidio of San Francisco. Try to clear. Um, is in the northwestern corner of the city, connecting to the Golden Gate Bridge. It was a former army base. It was transferred to the National Park Service in 1994 and has become a quasi-independent Presidio trust. Big part of it is the preservation of historical structures and forests. And there was a lot of unexploded ordnance and dumps that had to be removed, but that set the stage for ecological restoration of dunes, coastal scrub, grasslands, and riparian zones. And as part of the long-term ecological program, they're bringing back extirpated taxa. So here's just some photos of some restored dunes. I mean, literally, some of these areas used to be just, army garbage dumps and with all kinds of nasty stuff in them. And there's some really good stories that I won't be able to get into about like where did they find the sand to do this. Um, but there's been a lot of ecological restoration, and of course, it always starts plant communities. Um, but we found there were some opportunities to do some butterfly reintroductions of extirpated taxa. The first one was Euphydrias chalcedona, the chalcedon checkerspot. California bee plant and sticky monkey flower are great plants for restoring coastal scrub. Those are the two host plants. You can see the uh, checker spot larva on the bee plant here. So in 2017, we went to San Bruno Mountain, picked up about 1,500 of these late instar larvae, dumped them off in the Presidio, uh, did the same thing one more year, and then it totally took off. It is actually amazing to be out there. There are chalcedon checker spots everywhere. So by 2023, it had spread well away from the uh, release sites and just really, really worked well. This was a pretty straightforward one to do. Then we decided we would try reintroducing the California ringlets, Nympha californica. Nice mating pair here. Uh, its habitat's grasslands. It requires a mix of annual and perennial grasses like this purple needle grass here. So over several years, we went across the Golden Gate to the Marin Headlands and released adult butterflies in this area where you see the um, markings. And these were the iNaturalist observations from 2023. And that one continues. There's not nearly as much grassland as there is coastal scrub in the Presidio, so it's never going to get as abundant as the checker spot. But the star of our show, here's Glaucospsyche Xerces, the Xerces blue. It's a really beautiful photograph. I love how you can see almost every scale on the wings. Here's a female with typical dimorphism in lysinids, with the females being brown. One of the really outstanding characteristics of Xerces was the phenotypic variation on the undersides of the wings. So this is kind of the classic Xerces with with white spots but no dark infilling. Some butterflies had, the beginnings of black infilling, and then others really had quite a lot. These were named different forms. But I think it was one of the characteristics of Xerces to have this amount of phenotypic variability. So here's kind of a timeline, and we'll go into the details of each of these steps. So starting in the 1870s, we had the loss of the dune habitat in western San Francisco, development of residential and commercial spaces on the dunes. Now we have pretty densely packed houses in that area. The last Xerces were collected in 1941, and the last sighting was in 1943. Then and Bob Pyle started the Xerces Society in the early 70s, picked the name Xerces because it was the best documented extinction of a butterfly taxon in North America. Then in the mid 90s, National Park Service and the Presidio Trust were formed. And that's when the dune restorations began in the Presidio after it had been transferred from Army. Then in 2000, Bob published his Resurrection Ecology article. Well worth reading. You know, Bob just writes very well and very convincingly. Uh, then in 2017, the Presidio restoration ecologist Lou Stringer uh, contacted us to start doing some habitat, the you know, trying to resurrect the Xerces blue after the success of the Euphidius chalcedona project. Okay, then going back down, there was a query to the California Academy of Sciences about getting involved since they have the Xerces blue specimens. Um, a small amount of funding from a group, Revive and Restore, who are really into applying modern genetic techniques to conservation, funded the initial genomics work. Funding to do some niche modeling. I'll show you some of the results of that. In 2022, a really brilliant student who I believe went on to medical school. We got some funding from the Florida Museum via the Disney Butterfly Conservation Initiative. Jarrett Daniels really liked the project. Then we started finding places, including the UC Santa Cruz Fort Ord Natural Reserve on Monterey Bay and California State Parks. And then the translocation started in 2024 and 2025. You can see the marks on the butterfly so we could keep track of it. There's a nice photo of a couple of males and a female. Uh, and I'm going to tell you the story of this in a lot more detail. So, it's a photo of what Golden Gate Park in the western part of San Francisco looked like in 1910. Just endless sand dunes across the western part of the city. And then this is deerweed, the presumptive host plant of the Xerces blue. As we all know, the world was black and white back then. So uh, now here's a view of the former habitat of Xerces blue in the western San Francisco. You can see the Golden Gate Bridge up in the upper corner there. And here's Golden Gate Park. And the current present habitat of the Xerces blue is in cabinets in the California Academy of Sciences and other museums in Golden Gate Park. So, it's gone as a living entity. So our mission was to identify a surrogate for Xerces that could be introduced into these beautifully restored dune habitats. And we had three candidates of the silvery blue. Now we know Xerces was very closely related to the silvery blue. It's been kind of a controversy over whether it's a distinct species or a subspecies, but we'll leave that argument for later. The closest ones are subspecies incognitus. Then we have Sabulosa, which you'll see lives on along the coast south of San Francisco, and then Australis, which is in Southern California. So, which one is going to work? And then what's the best way to pick a surrogate? We can do the genomics and try to pick the closest relative from a genetic sense, or ecology and natural history, finding the best ecological match. So I'm going to go into those. So on the genomics, the first question is Xerces really a distinct natural group? And then what is the closest living group to Xerces? So, potential surrogates, as I was saying, because here we have Xerces, kind of the extremes of the phenotypes. We have Incognitus, Sabulosa, and Australis were the three candidates. We can see, different spot patterns on them, uh, named subspecies, and I just love the name Sabulosa. I call it Fabulosa, and you'll see why I like it so much for this project. So, specimens from the museum and some fresh collections were taken across California. The light lavender will always refer to the Xerces blue, but then we have the light blue is incognitus, very widespread uh, taxon subspecies. Then we have the brown is the Sabulosa from Monterey Bay area. And then Southern California, we have three subspecies, including pseudo-Xerces out on Channel Islands. So kind of keep those colors in mind. So a total of 69 specimens across this complex were sequenced. Not going to go into all the details of the sequencing and the analysis. Um, but we had 20 museum specimens of Xerces that were sequenced, and you're able to extract DNA from a leg. It's really amazing technology these days. And a lot of incognitus. We had one Sabulosa, and then a total of 55 museum specimens and 14 modern specimens. So that after all the genetic analysis, here's what we found out. So we came up with three natural groupings of the butterflies. So north and east of San Francisco, we had incognitus, and they all kind of fell into a similar group based on this very extensive genomic analysis. Then we had a Xerces, which turned out to be like really distinct at a genetic level. All of these are individuals, kind of fell in. There's like a little bit of potential other taxa in there, but stuff is. Then we go south of San Francisco, it goes back into the incognitus, the group. Then in Southern California, it's another distinct group. This is Australia's Palosaverdensis and Pseudoxerxes. And then in the Monterey Bay area, we have a subspecies Sabulosa that kind of falls, it's an admixture of incognitus and the southern subspecies. So that's basically the take-home story on the genetics here. And it turned out just barely the local incognitus were the closest relation to the Xerces, sort of a isolation by distance. So now we're going to go into the ecology. So what's Xerces' job? What's its niche out there? And then are there living relatives doing Xerces' job, be a stand-in as an ecological surrogate? So the difference between ecological differences between Xerces and silvery blues, um, host plant choice. They all take all the host plants are in the pea family, and Xerces was known to be especially on deerweed, acmosphon or lotus. And then the local Xerces, the local silvery blues, feed on a pretty wide variety of legumes from lupins, clovers, and Vicia, including some non-native introduced species of those. Then, of course, climate is really important for the butterflies. Xerces is in a cool, foggy location in Western San Francisco, kind of the definition of San Francisco's climate. And then the higher, much warmer, n less fog, and generally higher altitude. So we did actually, the student did a really good job taking locality data, overlaying it, overlaying it with climate data, and then building predictive models of where we get the best matches to the Xerces climate. Um, so it's a very multivariate kind of analysis. The lavender is the space in which all the Xerces uh, showed up. It's a very narrow range of climate because there's a very narrow geographic range. But then incognitus just a much, much wider range extending into warmer and drier areas. Then when we look at Sabulosa, uh, here it's like Xerces is kind of smack in the middle of Sabulosa in climate. Again, it's coastal fog belt in Monterey County down into San Luis Obispo County. And then for the Southern California, Australis versus Xerces, there's no overlap in the climate niche there. So we can get rid of Australis and the Southern California species, and not surprisingly, as a having a climate match. One other factor we weren't able to deeply investigate are the role of ants. In this photo on the right, there's a little ant in the middle of it because lycaenids have that symbiotic relationship with various species of ants. But we do find various lycaenid species in the sand dunes in the Presidio, and there's a pretty decent diversity of ant species. But this is definitely an area that needs a lot more work. Okay, so in summaries, we have the three genetic groups. They're all different. And the ones in Monterey, the Sabulosa, are sort of a admixture of the northern and southern subspecies. Yeah, the closest uh, genetically was the local incognitus, but they're so ecologically different. We decided we were going to go with ecological similarity because we have a cool foggy microclimate, have a demand, which is kind of important for both the host plants and then like where they diapause. Host of deerweed, and we don't quite know what's going on with the ants. So we went with Sabulosa as our choice. Here's a spatial representation with an oblique view showing the climate niches. The tan along the coast is Sabulosa that matches the Presidio climate much better than the more inland areas here. So we're really confident we got a good climate match. And, just to be honest, you know, before we even did all the quantitative work, it was pretty obvious that we wanted a sand dune dwelling coastal uh, subspecies that was on deerweed, just so we didn't have to have some big ecological change. Okay, so we used this map to go investigate a series of sites around Monterey Bay, down on the Big Sur coast. Yeah, this was what I'd call real harsh field work in one of the most beautiful places on the planet. And we went and we saw how many silvery blues we found, how much distance we covered on the searches, and then detections, butterflies per kilometer, just to identify places that had decent, large enough source populations that we could felt comfortable, A, we could collect enough to make it worthwhile for translocation, and B, we wouldn't be harming the local population. So back to the Presidio, it's like, what's the restored habitat look like? So we're looking at deerweed. Here it is in full bloom in color. Really beautiful plant. Here's some restored dunes at Rob Hill, and you can see all the deerweed clumps sprawling out. So we went to the various parts of the Presidio where dunes had been restored, estimated the number of deerweed individuals and also the deerweed cover. So up here at place Rob Hill, we had somewhere over 400 summed up deerweed plants. These other spots were larger and had more. So we had a good idea about what the habitat we were looking in. And then this is the percent cover calculated from the plant sizes. So, for example, up here in Rob Hill, we were in the 16 to 31% cover. So, one of the dominant plants up there. In some of the other sites, it was down below 3%. There weren't very many plants. So we want, a lot of plants and cover. And then we were also interested in, are the deerweed populations self-reproducing? So these are the size classes we had, 1 through 6, the diameter. So class 1 were basically relatively new recruits. Like a Rob Hill up here in the upper right, uh, 7% of the deerweed plants we encountered in the surveys were in the, kind of new plant class. So it was definitely self-reproducing population. Same thing at some of the other sites, but then there's one site, this place that has 140 on it, where we didn't have any new recruits, and it was actually getting quite thick with other dune species. So, but we do have self-reproducing deerweed populations in these sites, so we're not going to have to go in and constantly plant. So, again, just to uh, reiterate, the ecological surrogate are the Monterey butterflies because of host habitat and climate. But as an evolutionary surrogate, it's not the most closely related, but because it has this admixture of genes between the northern and southern subspecies, it has a lot of genetic variation. It might be more adaptable than other taxa. So our 2024 plans, we got the money from University of Florida, the Disney Butterfly Conservation Initiative. Thank you, Jarrett Daniels. Uh, so we decided we'd use ecologically similar Monterey populations. We would follow the releases with a visual mark recite, um, hence the Sharpie marks, to assess our success and then plan future work because this is going to be a learning process. So the release sites we chose, we have Rob Hill up at the top, we call it Hospital Dunes or Presidio Hills in the middle, and then Lobos Dunes. Interestingly, Rob Hill and Hospital Dunes are owned on land owned by the Presidio Trust. The Lobos Dunes are actually owned by the National Park Service. So in the first year, we didn't have a permit from the National Park Service, but we were free to work in the Presidio trust sites. So we worked at these two sites. So here's a view of Rob Hill. It's some adjacent eucalyptus forest that's going to be maintained because it's historic. It's actually occasionally used by monarch butterflies. Here's what the hospital dunes look like, very open dunes. An interesting side note here is that the sand that was used here came from the excavation of the parking garage in
Stuart,
Golden
you're
Gate
breaking up
Park
a little
under
bit.
PH.
Stuart, could you, if we could just pause for,
Yeah, oh, hi, yeah, it says my
you're breaking up a little bit. If you could
okay, so
just go
yes,
back to talking about the hospital dunes and where the sand came from.
okay, yeah, sure. Yeah, I just had a little unstable internet.
Yeah.
Okay. So anyway, yeah, so here's the hospital dunes. The sand came from the excavation of the parking garage underneath the Cal Academy of Sciences and the Young Museum, and it turned out it has a really high pH and is very resistant to invasion by non-native species, but the dune species did fine. And then here at Lobos Dunes, we're looking down towards the ocean to the left. Okay, so it's off to the races. We go down to the Fort Ord Reserve at in Monterey Bay, wander around with butterfly nets, grab some silvery blues, and then this is Darrell Capon, who is kind of the leader of the whole project with the Cal Academy, uh, driving the silvery blues up to the Presidio. Put them in individual cups in a cooler, and then the release was done underneath netting, so they didn't immediately fly away, and we had a chance to watch them, give them a little bit of time to acclimate. So on April 2 and 3, 2024, we gathered 15 butterflies from Fort Ord. I think it was 10 females and 5 males. We released at the Presidio Hills or the hospital dunes. They were released at 8 o'clock in the morning. We checked them again at noon, and 12 were still there. We noted some oviposition behavior, and then the mesh was removed at 4 o'clock. Going back doing the mark recite, we saw 6 uh, on April 4th, 2 on April 5th, and then April 6th through 8, we only detected eggs, which was still pretty exciting. Again, here's what the hospital dunes looked like. Here's one of the recited butterflies. And then here you can see the marks that were put on with Sharpies. Here's a different butterfly. And then, this is just showing the different butterflies were released and whether they were observed or not. So you can see, we only recited two of them after 2 days. But that's pretty typical. Butterflies are not long-lived and they do disperse away. So they stuck around for a while. Our second release, we gathered 28 butterflies around at Garrapata State Park in Big Sur and at Fort Ord Natural Reserve again on April 9th and 10th. We did the release at Presidio Hills, a slightly different section on April 11th. Same routine. Um, and here we actually had the resightings considerably later, up to 7 and 8 days later. So, Again, a lot of them just disappear or are not recited, and but they were around. And we also verified eggs. And then by April 12th or 13th, we verified that some eggs had been late. Uh, then we had some mortality. One was just cicated. It was a pretty warm day for the Presidio. One got nailed by a spider. Uh, One flew and may have been eaten by a swallow. So it's a perilous life to be a butterfly out there. This shows kind of the aerial view of the Presidio dunes and the kind of route that would be walked, searching for them. Here's this little alcove here was a good spot for them. I feel it's like a little more wind sheltered. So in that spot, we actually found a an egg, which I thought was pretty amazing because there's a lot of deerweed area to search. Here's another one of the resightings. And then close up, got two in one photo. These are two males. We put males in, even though the females were most likely mated, just to make the let the females know that they're in habitat, because one of the great truisms of butterfly ecology is that the best indicator it's a good place to be your species is the presence of other members of that species. So a lot of interactions. Yet another one. Durrell has a really big, long. We have a female and two males. Um, these are not in the same photo. These are all different photos. And then this was really exciting. Here's a female actually ovipositing. See the curled abdomen. Yet another marked butterfly. So we had three releases total, Presidio Hills, uh, again in the Presidio Hills, and then up in Robb Hill. We had released in May, we had released 28 butterflies, and five of those were recited. So, our preliminary conclusions from this first season is that the cooler weather improves survival under the mesh, but because they're not very warm, egg laying may not happen, and birds may be a problem. Warm weather increased the predation and desiccation under the nets. We have spiders and lizards, and birds are still a problem. We decided that moving nets off sooner than later is good. And then we decided that when we do it again, we're going to assign interns to each butterfly and track them over the course of day one, which may keep the birds away. And then it's important to give the butterflies time to find a good place, which may not be right near the release. And then the habitat choice can provide feedback on the release methods and potentially guide further restoration of the habitat. So 2025 comes around. We've learned a lot. So we moved over to Lobos Creek and the Lobos North Viewshed. Did this on Earth Day, which is just great symbolism. We always like that butterflies are free moment. And up to five were seen afterwards. Then we released another 10. And most importantly, we cited offspring from 2024 females because all the new butterflies were marked. If we found an unmarked butterfly, we knew it came from the previous year's releases. So on April 12th, we found a male, had a different male on the 25th and 26th. Then we had a female and a male at Robb Hill, and then we think it was the same female at Robb Hill. So we actually got successful reproduction. Um, So our next steps for spring 2025 and beyond, we want to continue monitoring and follow up and do follow-up translocations. Uh, it often takes many translocations to get a butterfly population to take, ensuring some degree of population stability by upping the numbers and avoiding Ali effects, things like the butterflies aren't finding each other, so they take off. Um, and then a lot of outreach efforts and community engagement to support long-term success. So Liam O'Brien made this beautiful poster for the project by the light of the silvery blue. And then we put up these placards, signs around the Presidio at the sites, uh, just explaining the project in a relatively short period. Um, there's a QR code that you people could point their phone at and get a lot more information. And this is we got a lot of so uh prior to the project we had some then during the release period in April and May 2024, we had two plus, six media hits, and that trailed off into the summer. The second year was just kind of crazy. Durrell was like all over the radio and a lot of print, including like Bay Nature magazine, um, and continued on. So this project generated a lot of interest. I've made the Washington Post and some national news. They did a kind of media outreach analysis. So there were in 2024, there were 248 mentions of the project. And of course, the estimated reach of that is, potential, 800 million people, and that got global coverage in 10 countries. And it's always positive reception in all three times. And in 2025, it was fewer um, mentions, but we really saturated the media with this. It was very exciting. Maybe I can send a list of the various um, media outlets so you can get a sense of what people were writing about it. Yet, the future funding on this project remains elusive. Everybody loves it. It's a really cool project, but we have not been successful getting funding moving forward at the type of scale that's really needed to do this, which is, very disappointing. There's going to be some translocation work done this year using internal funding from the Cal Academy and the Presidio, but it's not going to be at the scale that that we need. This stuff costs money. So, and this is made especially bad by the loss of federal funding for a lot of conservation work. Fortunately, the Presidio is quasi-independent, and they fended off an attempt to steal their funding very early on in the current administration. So, a lot of groups and people were involved in this. And again, I really want to thank Durrell Cappan of the Cal Academy for you know basically spearheading the whole project once we had identified the potential for the habitat in the Presidio. And that's it for my presentation.
Okay, well, thank you so much, Stuart. This is really a fabulous project that you're working on. So now is our time for open questions. I have several, but you all just chime in if you have questions. Well, let me ask a question to start it off. It's kind of an obvious one. Well, first of all, does the increased media interest translate to any additional funding? Uh,
I wish we're trying
but you
to,
don't see evidence of that?
yeah, but we don't see evidence of it
Okay.
yet.
And then the second question is, what's the role of the Xerces Society?
I should speak to that.
Okay.
Yeah, I'll let Bob speak to that.
It's a bit surprising, with that they were named for this butterfly and yet they're not a sponsor. So it's
It certainly is. I'm glad you asked. Do you mind, Stuart, if I take that?
Bob, please go ahead. I don't want to speak for Xerces.
I've got a couple of other comments too and questions, of
Yeah,
course.
please. And it's open discussion. Feel free to unmute, ask questions. It's open discussion.
Let me say first that this is about as gratifying as you can possibly imagine for me to have written this essay 20 years ago and seen in 10 years, took 10 years for anybody to really pay attention, another 5 for them to start talking about it, another 5 for it to happen. And that's pretty fast, really, and that's extremely gratifying to go down there this past spring on the train and go out there with Stuart and with Durrell, and Mia, and to see the butterfly. Well, I didn't see the blues that day. They saw them the day before and the day after. But we saw Akmon blues, and we saw the chalcedons, and we saw the other things. But the blues were there, and to be able to see it happening, extraordinarily gratifying. Second thing is that you might recall from my talk last month that the inspiration for me for the article, the essay, in the first place, was the English experience with revivifying the English large blue that had become extinct in the 70s after 50 years of trying to conserve it. Jeremy Thomas finally figuring out how to manage the habitat, getting the habitat in shape, and then going to get a surrogate. But in this case, the species was not extinct. It still occurred in Sweden. So they could bring back a 10,000 year removed um, replacement from several different sites. So they got genetic diversity, plunk it down, and it's done extraordinarily well. Now, that one had much more complicated ant symbiosis, vastly more complicated than Xerces has. So that was a difficulty, but it also was the same species, which was a bonus. But that's what gave the urge for this thing. The other thing is that um, I forget what it was. I may come back later. But let me get to your question about the Xerces Society. So when I got this idea and the term resurrection ecology, I first published it, I think. I published it in 3 places. And I think the first one was in the Xerces magazine, Wings. Now, my role in Xerces for many, many years has been solely as an advisor. I'm just the grandpa. As soon as we've hired professional staff, which was first in 1985, I resigned from the board so I wouldn't be, a founder thorn in their side. I learned from David Brower and others that Glassberg that doesn't work to try to stay the king of the organization forever. So I've just been an advisor ever since, have no undue influence or certainly no authority in the organization. So when the article came out and when really first it was Nick Grisham in Texas, the genomicist, who wanted to sequence the Xerces blue for his sequence of papers on butterfly genomics in the US. And I went to the Xerces director, Scott Black, and I said, look, if we could kick in a little bit for this, we could we could finally find out what Xerces consists of genomically. And maybe we could start looking forward to a reintroduction. But even though they had published the article first, Scott felt that a number of the Xerces members wouldn't like the project. I think they thought it was going to be more like the dire wolf woolly mammoth, dodo kind of stuff that the revive and restore people are into, and the
Mm,
what's the other group called? Gigantic?
mhm.
That's not it. The people who have spent millions on the mammoth and the dire wolf.
Yeah, Mammoth Biosciences or something.
Something called gazillion
Yeah,
or
anyway, yeah.
gigantic or something like that. Anyway, um, so he thought, and he said that they'd never had more complaints from the membership about anything they'd ever published than they did about my article. Somehow it got up some people's noses. Now, it was also published in Wild Earth and one other place. And the interest was always very positive. But there were some people in Xerces who found it too tinkering with nature. Um, and Scott Reed finally decided that they wanted to spend their money instead on direct conservation of things that are already there. And that's fine. I haven't tried to argue with them. I understand their point of view. But the fact is, I have not shrunk from reminding them of this a little bit. That when that media frenzy occurred in California, I would occasionally send the PR pieces to Scott and say, "Hey, Scott, you could have been in all of this. You could have, put a little into it and possibly gained a great deal from it because there are an awful lot of people interested in this." And I find it a little bit embarrassing, but surprising and puzzling why Xerces has not been able to see that, as their namesake, that they should be deeply involved in this in the first place. But as I say, I have no influence other than making suggestions, and that has been their decision. So be it. But that's why Xerces isn't in there. I think it's a mistake, but that's just me.
Yeah, I think early on there was like this with the revive and restore people, there was like, yeah, we have all this technology, we can like insert Xerces genes, and then it's kind of like, we're just gonna go ahead and try and figure out if we can like get a silvery blue population established. We're not
Well,
gonna wait. We're not
exactly.
gonna wait until, you figure out like how do like what genes should get spliced in to create that spot pattern.
Yeah,
It was just,
that unsatisfactory replica. And I said something about this last month, but we talked about this, Stuart. That article you asked about that was, and might still do, it was simply saying this. Um, those people, what's the name? Anybody here, somebody here must know. Anyway, there's a big New Yorker piece about the dire wolf.
oh God. Oh
I forget the
God,
name of
that
the
was painful.
Yeah, I know the name of the organization. I can't think of it. But anyway, um, they have created these little cute little puppies with big jaws and white fur and a couple of traits of the dire wolf and absolutely no habitat for them. And they're not even dire wolves, long way
Yeah.
from the 904 that have been pulled out of the La Brea tar pits. But
Yeah.
and they've spent millions. And they said to the reporter in the New Yorker that they expect to make millions off of this. I mean, that the bringing
Yeah.
back the species is not their main thing. Their main thing is making money.
Well,
Well,
I also was really amused by the fact that their phenotype for the dire wolf came out of Game of Thrones.
well, good point. Good point.
You it's like TV reality or TV unreality. I
That's
mean, yeah,
pretty much
everybody I've talked to who knows anything about this is just like, what the?
that's pretty much what it comes down to. And so I like to say, compare this. This would be, if I write this article, this would be the point. They've spent millions to get these poor cute little creatures that the New Yorker concluded might be the first example of a double extinction. They became extinct once, and now because they got no place to go, they may become extinct again. Whereas we here in the Presidio, and I say we, I mean all of us,
Yeah.
the citizens of the United States who are the owners of the Presidio in absentia with the Park Service and so on, we have a darn good surrogate for the Xerces Blue. We can go there and see a near distant evolutionary relative of the Xerces boo flying and doing, I love the way you put it, Stuart, doing the job of Xerces, which, like the English large boo, over time, under the same environmental stresses and opportunities, may begin to microevolve towards something Xerces like. And if it doesn't, it does no harm and it gives people joy. What could be wrong with that? Now I want to ask you a question, Stuart. How much? How much do you need?
Oh, um, I'd say, minimal 25,000 a year, or maybe 50,000. We can always use more.
And that's nothing. So you look at these people who spend millions and they got
Yeah,
nothing really. And you've,
yeah,
I think Durrell said under 100,000 so far, which has leveraged
yeah,
millions for habitat
yeah,
conservation. And you need
yeah. And
you need
I
25,
mean,
50,000
I,
bucks a
I,
year.
I think, I think when you add in the in-kind staff time, it goes up. But,
Sure.
that's what people are being paid to do anyway. So,
Okay, so let's all get busy and try to think of ways to bring together a few grand for this project. It definitely deserves it.
yeah. Okay.
Colossal biosciences.
Colossal, that's
Colossal.
it.
I knew it was something like that. I mean, a real nice, modest name. No, don't get me wrong about Revive and Restore. They're good people doing some good things, but
Yeah.
they do feel that the genetic manipulation might be the way to go. And frankly, it's a complicated, it's not Occam's razor, I'll tell you that.
Hmm.
No. Are they at all interested in providing some funding? I understand they provided some
They did up front,
technical
which is
expertise in particular, but I mean, it seems like they might also be interested in putting a modest sum of money into this project because it's a different approach, and it seems like they might value that.
Yeah, I mean, we're definitely like using the genomics to, we're not talking about trying to do any kind of genetic manipulations here. We're just trying to get little silvery blue butterflies flying in the Presidio again. Just
I
makes
also
it
love the
makes
verb
it a
you
richer
used.
place.
You used a great verb, a great phrase. You said something about we brought him back and plunked him down. That's not quite what you said.
Oh,
What did you say? Tossed him in there, plunked him
uh,
down.
yeah, we'll have to look at the
Or
video, something.
Because you see, that's what we want
Yeah.
the Fish and Wildlife Service, the feds, to finally get off their butts and do with the Oregon silver spot in Washington. They got the butterflies, they got the habitat, and they go meeting after meeting after meeting over 40 years now, and they can't just decide to plunk the butterflies down on the Washington side of the Columbia River.
Mm.
And so thank goodness you didn't have to deal with, and forgive me if I sound um, a little bit reactionary here, but that you did not have to deal with federal regulations. And by the way, if you had chosen uh, Palos verdiensis,
Oh,
Palos,
that would.
Palos verdicensis, which is a silvery subspecies in Long
LA.
Beach, yeah, near Long Beach, that in Rancho Palos Verdes is a federally listed taxon. Not only that, it was thought to be the very first entity that went extinct under the federal um, um, protection because after it was listed, it was thought to have become extinct. The very first loss under the ESA. But then Rudy Mattoni rediscovered it, started breeding it big time, and so it still lives there at that
Right. So it's
and
like a naval
naval.
industrial
It's
complex.
it's a naval munitions and gasoline depot.
Well, and, I've talked with like Travis Longcore about,
Yeah,
and the problem is to renew the deerweed populations, they need to disturb it, but they're not allowed to disturb it because that might result in the take of the individuals.
yeah, exactly.
So,
So thank goodness you
you
didn't
know,
have
uh,
to deal with.
my yeah, my paradigm for butterfly conservation is we need to restore the habitat to the point where the awesome reproductive powers of these butterflies can be expressed
Right
and
on.
not worry about every last little individual,
So thank
you know.
goodness. I mean, I
That's,
love I'm a Democrat. I believe in federal regulation, okay, but not
yeah.
sometimes when Fish and Wildlife gets in their own way to get anything done. And that was the case there. I'm glad
Yeah,
Paul
that's
Sturgis is still there, but thank goodness you didn't choose
kind
that.
of the same, you know. Well, yeah, yeah. Well, fortunately, it wasn't a good climate match.
And one other question,
And
and then I'll
yeah,
shut
so
up.
okay.
The subspecies Pseudoxerces in the Channel Islands. A lot of people said, why don't you use that? That's the one that actually looks most like Xerces. In fact, it has some of that same kind of variation of the um, uh, white spots with or without irises. But I think that's just a matter of convergence, right? And as you showed, it's not an
Yeah, I
ecological
think
match at all.
so. Yes. Yeah, some ways it does have some ecological similarities, but you know, you actually need some large, extensive populations to be able to take butterflies for
Oh,
translocation. I
right.
mean, it's kind of self-limiting. If you have a really diffuse population, you can't collect enough to make it worth it,
Yeah, plus it wouldn't be
you
easy
know.
going out to the Channel Islands to try to get your
Yeah, yeah,
stuff.
yeah.
I'll shut up. I'm sorry to talk so much.
No, so
Uh,
anybody else want to unmute and ask a question, make a comment? Some of you, habitat, yeah, Mary, go ahead.
Oh, hi, Mary.
Hi, Mary.
Yeah, hi Stu. How you doing? Good to see you.
Good, good. I was kind of
Hey,
hoping you'd
I'm
show up.
curious to know you've got a couple of different. Oh yeah, yeah. You've got a couple of different sites there that you started reintroducing at. What's kind of your plan for the number of sites and the number of translocations you think for each of those sites over
Uh,
time?
um,
You've frozen for a moment, Stuart.
so I oh okay. So we have, you know, my internet connection is unstable again. So we have three sites. I think I'm not sure exactly what our plans are for the next round. One of the exciting things is, is that some of the senescent stands of Monterey cypress are getting that's actually going to connect the Lobos Creek with the Presidio Hills hospital
Mm.
dunes. Moving targets, Lou Stringer is just the master of dune rest. I, that's a really good question. I don't know what the plans are for 2026, but we're going to probably reinforce populations that we've already
Mhm.
uh, sort of established. You know, it is really nice not working with listed species.
Right
You
on.
know.
Yeah. Yeah, I mean, one of the things that I really love about the project is that it's the kind of work that needs to happen on a lot of different scales and a lot of different places. I know you're familiar with Taylor's checkerspot and how we have kind of holes in the range where we don't have any kind of
Mhm.
suitable, local population, and it's a great model for how we can try to fill those gaps, right? I think the typical approach usually is to try to look at the genetics, but it's not necessarily always, really the most pragmatic approach, right? So I really like
Yeah,
what you did with this one.
yeah. You know, to me, like it became really obvious that Sabulosa was going to be, you know, the Fabulosa uh, uh, target. Just, it's kind of the same habitats. Everything matches up, and asking the butterflies to like switch host plants and be in a cool, foggy habitat when they come from a much warmer site. It's just like, no, it's too much of a stretch.
Yep.
Yeah,
So,
I had not thought of that when I wrote the piece. I was thinking of genetics, but that makes all the sense in the world. And I was very impressed with the modeling that the young woman did
oh,
on the ecological
yeah.
concurrence there. That was very impressive. And I think you made just the right choice there.
Um, sometimes the answer just becomes quite obvious when you think about it. Yeah, just talking about getting in their own way. I've been involved with the Lang's metal mark, and that just disappeared because they just couldn't. They tried captive rearing and releases. It didn't really work. The habitat, there's a lot of issues with the habitat. But we are trying to get some, the goal is to get some form of Apidemia mormo established on the Antioch Dunes. And we think our idea is just to go to some of the nearest mormo populations and just translocate adults
Yeah,
and mix them all up and hope that the genetics work out.
it should, if the habitat's still there. At one point, it became almost a monoculture of gnomes, and people were worried. Jerry Powell thought that all the attention to Lang's metal mark was letting all the other dune insects
Mm.
uh, be destroyed. They have
Yeah, well,
millions
I
spent
mean,
there, but it never really worked out.
it's like a 55 acre site, so there's like
Yeah,
only so much you can really really do on it. And
with
the
so
last
much
remaining habitat was getting dusted by a gypsum dust from a gypsum plant.
right next
And
door.
yeah, yeah. So and a fire took out the main population too. It's
Yeah,
just
yeah. I'm going to say one more thing. I said I'd stop. Stuart, I wanted to thank you for your very kind words. But I also want to say if anybody wants to read the original essay, Resurrection Ecology, if you just send me an email, I'll be glad to send it to you.
oh, okay. The
All
the
it has,
one in Wild Earth.
pardon me, yeah, the one that was in Wild Earth and
Yeah.
Wings, right?
Actually, I found it online. So
Oh, okay, you can easily do that. Yeah, just
yeah,
that's probably easier. Just Google my name and the title Resurrection Ecology.
yeah.
It'll be there.
Oh, any other questions, comments? Okay, well, I have a question. I um, if Incognitus and Sabulosa interbreed, was there any consideration of introducing more than one subspecies?
Hmm.
Um, we were just really focused on the source populations being as ecologically similar
Mhm.
as possible. Um, yeah, I mean, there's subspecies they might interbreed, but it's kind of ecotypic variation that it's probably not a good idea to mix it up so much. I don't know. It just became really easy to focus in on the sand dune
Yeah, I
dwellings.
completely understand. I guess I was just thinking about more genetic diversity, more, different genes that would be brought into the mix. And so, but I
Yeah,
understand that, keeping it simple, there's an argument for keeping it simple too.
yeah.
Okay, any other
Okay.
comments, questions?
One comment I would throw out on that is, I know, Stu, this is something you kind of tossed my direction when I was struggling with some of the same stuff, is the whole idea that, these butterflies really are, as they're establishing, they're also self-selecting for adaptations to that habitat. And, it's not like you couldn't consider another subspecies down the line if things are not adapting very well, but I like the kind of singular approach and seeing where that goes. And that's where I think the work that's been done, the background work that's been done, is a really good framework for, trying to take your best guess at who's gonna survive and then see what happens. So, and it is pretty remarkable how quickly things do change. I think, generations are only a year or maybe even less, depending on the species. And so you can whip through a lot of generations and a lot of adaptive change in a fairly short period of time.
I think maybe the biggest adaptive bottleneck might be just we're putting them in postage stamp sized habitats, and all the ones that have a high dispersal tendency are just gone from the gene pool. And whether you can win that race with selection, I mean that's what happened with a bay checkerspot reintroduction attempt. We attempted, many years of bringing in like thousands of larvae from a big robust population, and it's just from like a site that covers thousands of acres to a site that's 40 acres. I just think there was too much leakage, and the selection for, and I call them flower potatoes, the homebodies, just was not fast enough. And we had some just horrible weather years. I don't know whether you had the issue in 2015 with like the worst butterfly year ever. It was like it was a year with no snow in California. It was just really bad year. And then also more
2020 was
recently,
down our worst.
yeah, I mean more recently we had the hellacious heat waves. The you got with checker spots, you got their diapausing in shallow soils, and then when you get 115-degree days at one after the other, and that doesn't do anybody any good. So, we with the bay checker spot, we had big population crashes from 2024 to 2025. And we had like a record heat wave in late June and July 2024. I think it just got too hot for them and diapausing in the soil.
We don't tend to have that kind of heat up here, but we had heat dome a couple of years ago.
Yeah,
It had a big impact.
yeah, and I
Yeah.
I remember hearing that really hammered the Taylor's populations.
Absolutely, yeah.
Which Mary's worked on a lot, of course.
Yeah, 2021 was hands down the worst year, and it's like it was even before the heat dome, because we really didn't have any rain in March and April that year. And so not only did we lose the butterfly, for the same kind of reasons, just kind of cooked in place, but it destroyed all the host plants as well. And so we didn't even have the habitat for them to come back into. So it's been pretty profound. And, it's like we're just slowly starting to rebuild from that. It's pretty impactful.
And I liked what you said about the what did I like? What did you say? Oh, shoot.
But
Carry on, children. I'll come to it in a minute.
Where do your volunteers come from, Stuart?
Oh, they were from the Cal Academy. They have interns. One of the things I've found is that when we get interns involved in these butterfly translocation projects, sometimes it's like their first real experience. And I just tell them that, man, it doesn't get any better than this, just this is the coolest thing you're ever going to do. You might find something as cool to do, but you just stepped into like the most fun, interesting project. So enjoy it while it's here.
Yeah, we caught your irony about the terrible fate of having to go catch butterflies at Fort Ord.
Yeah.
Yeah, terrible,
Yeah,
terrible, terrible.
overlooking the Pacific Ocean.
Yeah, It's like when Mary Linders and I were down 10 years before that, down watching cool squirrels in El Salvador. It's not a bad way to spend your time.
Yeah.
I remember what it was. Jack Dempster, who was the professor of Jeremy Thomas. Jeremy Thomas, who was now the Hope Professor of Ecology at Oxford and an OBE because of his work with the large blue. He was just a grad student at Monk's Wood at the same time I was when that Xerces came out of. But his teacher was a guy named Jack Dempster, and he was one of the first great ecologists working on the particular ecological needs of a given organism in a given place, as taken separately from the synecology or the whole area ecology. So Jack was trying to restore British swallowtails. So only one species they got. It's called the swallowtail to Wick and Fen, which is where Darwin used to cut glass from Cambridge and go catch beetles and swallowtails at Wick and Fen. But they become extinct during the war, World War II, because of changes in management of the reeds. Nobody cut the reeds for thatch during the war, so it all grew up and to willow car, and it covered the uh, Pucedinum palustre, the umbel that the caterpillars fed on. So Jack was trying to put them back, and they got the plant back, they got the reeds back to the right, but their source of donor butterflies was the Norfolk Broads, the only other population of swallowtails in Britain. Well, the Norfolk Broads are big and broad, as it says, and they were butterflies that were used to flying around. And so they brought them back and dumped them down in this couple 100-acre National Nature Reserve. And of course, they all flew away. So what you said about the um,
Yeah.
um,
Flower potatoes.
exactly, that was such a wonderful term, the flower potatoes. That's what you need. You need flower potatoes instead of the vagrant individuals that are going
Yeah.
to come from a widespread population.
Allison,
Yeah,
did
I
you
think
have a
Mary
comment?
told me that was a big issue at oh God, I'm forgetting the name of the site. Um where you release tellers at uh, it's a place,
Where
yeah,
they just where they dispersed, you mean?
Prairie Appreciation Day. That
Oh
that site.
yeah, so yeah, that site. I think the issue there was the area where there was actually host plants was really limited, but there was,
Yeah.
800 acres of, nectar, right? And
Yeah.
so how do you find the host plants in the field of nectar?
Hmm.
So I think
Hmm.
that was a big issue there for
Yeah.
sure.
Which site was that, Mary?
Hmm.
Glacial heritage.
Yeah.
Yeah.
Glacial
Beautiful when it
heritage,
blooms.
yeah.
But
Oh
very, I
God,
think
they're
very challenging
gorgeous.
site too because of, the swales were both hot and cold, right? They were really hot in the summer. They concentrated a lot of heat, but then I think they were cold sinks in the winter, so it wasn't great for the larvae in February. And then, that on top of the, very distinctness of the patches of host plant. So yeah,
Yeah.
tricky site.
Does
Yeah.
butterfly even occur there now?
No, no, we went through about 5 or so years of releases and they just didn't really take. And so we left that site. They're continuing to restore it and work in other parts of the site. We're trying to build on what we had in terms of habitat. But we moved on to other sites that, were more promising. So
Is Mardon there?
no, no, there haven't been any modern reintroductions at all. So unfortunately, it's like we're really,
Yeah.
really
I mean,
late on that one. Too
I
late.
think I think we're finding putting Humpty Dumpty back together again is difficult,
Yeah.
Yeah, easy.
Well,
especially
and
with a rapidly changing environment. It's just
yeah,
challenging.
I think it begs for the need to act sooner than later. I mean, it
Yeah.
was always hard to wait till the end and make it happen, right? But
Yeah.
the more we let the habitat fall apart, we don't know how to put it back together. And then we're, trying to reintroduce one novel thing on top of another novel thing. And it's the bar's
Mhm.
too high.
So
Yep.
Allison, did you have something you wanted to say?
Yeah, I noticed that you had released the checker spots as larvae, but the blues as adults. And I was just curious how you decide when to release as
Oh,
caterpillars and when to
be,
release as
okay. Because you could literally go to San Bruno Mountain and just like pick larvae off of Plantle. And then, the other aspect of it is we're actually bringing in a more complete food web. So we're bringing in whatever parasitoids are there. Um, so yeah, they're just a lot easier to handle by the hundreds or thousands. And then it's really hard to find the silvery blue larvae,
Well,
you
and
know.
the larvae are
So
flower potatoes, so that's an advantage.
yeah, they're also really cryptic.
Mm.
Whereas checker spot larvae just sit out there and bask. And it was the same thing, like we did adults with the ringlets just because there's a lot of ringlets flying around up in the Marin Headlands. So that was kind of an interesting thing about permitting is that the state of California is asking for permits to do these kind of movements, except for the um, for the ringlet, we were going from Park Service land to Park Service land, so we didn't need a permit from the state.
Okay. That's great.
Yeah, like dodging and weaving through the bureaucratic and social landscape is often more challenging than the biology side of it.
Yeah, well, the state of California won't even allow you to touch a monarch butterfly right now. I mean, I'm sure you have
Yeah,
permits.
yeah, I don't touch them. I just analyze the habitat.
There you go.
Yeah, but it's really exciting that tagging they've been doing is
Yeah.
really check out Project Monarch. It's really worth checking out.
Are they starting to use the radio tags?
Yeah, yeah, they've this is the first year and it's just yeah, it's amazing. It's Project Monarch is the name of the site. And then um yeah, so the Thanksgiving count or mid-season count as they call it now is going to be in at around 10,000 again. Another lousy year. Didn't recover from last year.
Does Project Monarch deal with both the eastern and the western uh, radio
Yeah,
tags?
yeah.
Yeah,
I,
David James has been involved in Project Monarch, and there's talk about getting more involved in it. So I don't think he's on here. I think he jumped off a little bit ago, but he's been very engaged. Melanie knows a little bit about it too. I don't know if you want to comment, Melanie.
Well, in the absence of Melanie, I went like maybe the most exciting part of it was they had a butterfly left Santa Cruz and flew like over 100 miles down the coast to Cambria.
wow.
Yeah.
David James, I guess, has jumped off also.
And then
Yes.
also like, I think the most valuable data might be when they migrate from the over and when. 'Cause that's the biggest data gap we have.
Say it again, Stuart.
Oh, when they emigrate from the overwintering sites,
Yes,
where do they go when?
the Californian one.
The kind of yeah, California, yeah.
I agree.
So
Do you want to offer anything about Project Monarch, Melanie?
Project Monarch.
Yeah, we were just talking about it. Yeah, I think you
it's
stepped
I'm
away.
sorry,
the
I was out of the room for a minute.
That's okay. Yeah.
yeah, we're just talking about the radio tags, the Bluetooth.
Yes. Now, I don't know much other than having read about it. So I'm sorry.
Well, and David James is getting more involved in it, and so WBA may be getting involved in it here before too long as well. Come spring, yeah.
The surprising thing to me was that it's not like we always pictured it, that eventually there would be radio tags that could transmit to a satellite and back down, and you could track them from afar. That's not how it's being done. It's being
No,
done
it's a
with,
little
with the frequency of cell phones all along the
yeah,
way.
little iPhone app on Bluetooth.
And so what
Yeah.
it depends upon is the frequency, the distribution of watchers with cell phones instead of long-term transmission.
Yeah.
Hmm.
Yep.
So, any other questions? I still have another one or two, but I want to give other folks a chance. Regina or Karen, you folks have been involved in some of this work, or do you have any comments or questions?
No, just that it's a really good presentation.
Hmm.
Oh,
Yeah,
thank
real
you.
interesting.
Well, you're an easy audience.
Very enthusiastic. I have a
Yeah,
question
yeah.
about the Sabulosa and ant interactions, whether there has been any work on that.
No, there hasn't. It's one of those areas we'd really like to get some students going on. Just seeing like what are the ants on the sand, on the sand dunes in Monterey. Um, and then doing a really good, thorough ant survey of the Presidio. We have a species about their distribution and abundance. It's really common.
I just wondered if there was any work at all on Sabulosa ant. I understand, not in Presidio, but if there's
Yeah,
been.
I not that I know of. Um, the most recent ant lycaenid work um, was done at Jasper Ridge on Ackman blues and Argentine ants and other native ants. They actually found that the Argentine ants would tend the larvae. I don't know. It's just, nice juicy secretions.
Yeah. The thing I mentioned about the English large blue uh, being much more complicated is that genus Maculinea, which is an old world genus, uh, has an obligate relationship with ants, where
Yeah.
they have evolved such a baroque fashion that the that the larvae, after feeding on wild thyme, in the case of the British large blue, THYME, drops onto the ground, is picked up by an ant, which is Myrmica sabulosa, I think,
Oh,
or
oh,
Sabuleti, Myrmica
yeah.
sabuleti. They both mean sand, of the sand,
Sa-
um,
Sav
and taken into
Sav
the nest. And the ant larva, sorry, the butterfly larva, in I
eats.
think the third instar, becomes carnivorous on the ant brood.
Yeah.
And the ants continue to milk it for the honeydew and tolerate it. And then all kinds of exchange of bogus pheromones takes place for them to pupate down there, hatch, and crawl out. So it's absolutely obligate. They've got to have the ant. The ant doesn't have to have the butterfly, but the butterfly has to have the ant. And that made the whole reintroduction thing so much more complicated because you had to get the right species of ant.
Right. You had to get the right species of
Yeah.
ant, and it had to do with the height of the sward,
Yes.
which
And the
created the
sward.
microclimate. So
Mm.
you and it was because the they took off the sheep grazing, and then also the rabbits got killed off by
Rabbits got
the
killed off by
myxomatosis. Yeah,
myxomatosis.
it's just so convoluted.
Absolutely.
Uh, yeah.
And so you had to first understand it. Robert Gooden was the first to really kind of get it. And then Jeremy got it scientifically, and they finally got it where they could put it all back together and get
Yeah.
the right species of ant. Because the wrong species of ant would pick up the larva and, of course, eat it. We don't have any of that because for most North American lichenids and ants, it's a facultative thing. They can take it or leave it. I don't know if any of them are actually obligate or not. Do you know that, Stuart?
I don't know. I mean, I know, a lot of evidence that it helps ward off predation and parasites,
Right.
parasitoids,
But I
but
don't know if it's ever absolutely necessary for any of them. Pillman might know, but he's not here tonight. But I'm not sure. But that particularly extreme coevolution has, and that's always the case. The more complicated that ecological needs become, the more likely a species is going to become threatened or endangered because it's easier to bugger things up.
yeah, sure. And then when reduce the available habitat to like a fraction, a percent or less of the previous extent, I always ask people to like imagine the ancestral condition of a lot of these species. There's habitat everywhere. And you have like really robust metapopulations, and that something could go extinct here, but then it readily get repopulated or rescue effects. But now, when we get stuff reduced to postage stamps, it's like you totally lose that. So some, like with Taylor's checker spot, just imagine the full glory of the prairies up there, being maintained by the indigenous people. And now we're kind of dealing with these tiny little patches that are left.
Yeah,
5,000
it's
years
actually
ago.
an interesting question because it's one of the things where even in like South Sound here, we have big patches, right? Compared to what Karen's got up, in the Olympics or what you see down in Oregon. And that's been sort of an interesting question is, when you have big flat areas, right? How do butterflies kind of find that habitat diversity, that microclimate diversity that might be the key to them surviving year in, year out, right? Might be this big flat spatial equation that's different from where you have a lot of, verticality and your microclimates vary with
Mhm.
elevation and, aspect, right? And so that's been, one of the things that I've really wondered about here is do we have to have bigger populations of checker spots here in South Sound and big prairies to support them? That differs from what you need in the mountains where they can maintain smaller populations because they've got, more diverse microclimates.
Yeah.
Good point.
So Vivian, you're welcome to unmute and comment. I guess I'm kind of curious, since you're in the area, if you have any thoughts or comments. Don't feel pressured. I just wanted to invite you to speak if you wanted to.
Hi, sorry, I was actually in another room away from my
Oh,
computer.
okay.
I'm watching it on my TV in a different room.
Oh, okay.
Uh, uh, no, I was excited to hear the talk. I didn't know a lot of what Stuart just went over, so that was great to hear about it.
So living in the area, are you inundated with these stories or not so much? And how do you know about this work? And
Well, I actually do a butterfly count at Jasper Ridge, which is where Stuart has done a lot of work. When I became a docent there, we started a butterfly count that we do every week, which was reproducing a work that had started in 2006 by a postdoc. And so for the past 10 years, I've been doing that with the partner that I started it with in 2016, I think. So we're almost going to be at the 10-year mark
Wow.
this coming year, which is fun. We go out every week to take a count on a bunch of transects across Jasper Ridge. But yes, a lot of these stories, in fact, I was also looking at a book that just came out recently by Liam O'Brien, which documents a lot of the butterflies in the Bay Area.
It's a
and so and
beautiful
he
book.
talks about a lot of these stories. In fact, on the pages about the Euphrates Chalcedonia, he actually mentions there's a restoration, that started in 2017. So
Yeah,
yeah.
Liam did. Here, hold on a second. I have the book right here.
Oh, good. You
Well,
might want to get
Vivian,
him for a program, Paulette. Liam's great.
Here, oops, sorry. There we go. Okay. There we go, sort of in focus.
It's from Heyday
Anyway,
Books, and it just came out a couple months ago, right? 2 or 3 months
yeah,
ago.
yeah, it's really fun, and boy, Liam is such a hoot to be with out in the field.
He really is.
Character.
Does he still do about 12 or 15 annual butterfly counts?
Yeah, I'm not sure. But we've been doing a lot of translocations of mission blue butterflies that have been really quite successful. We repopulated Twin Peaks, which is the type locality. And that was Liam, who was the instigator of all of that. But now a lot of habitat restoration, diversifying the lupin instead of just primarily having one species of silver lupin. The other two species, the varied lupin and the summer lupin, are really key because the silver lupin gets hit by a fungal disease every 5 to 10 years, kind of melts down. Yeah, so that's really good lesson on biodiversity.
Couple of other Liam's other projects in the city have been the Green Hair Streak
Yeah.
project. He discovered there was a butterfly that at one point was thought to have become extinct in the city, but it didn't. The green hair streak. It's related to our bramble green hair streak, and it's gorgeous. It's subspecies viridis, meaning green. Just beautiful green hair streak. It's right in San Francisco, but it lives on a species of
Buckwheat.
buckwheat of Gnum, Eriogonum, and he got the idea that this buckwheat is here and there in little bits, and if we found all the different little patches at street ends and intersections and and slopes in the city and got the Eriogonum going there, green hair streak was spread. So he promoted this all through the Mission and the Bay Bridge. What's the neighborhood up there on the west? Stuart, where you
Westport.
oh,
Yeah, Corona Heights.
yeah,
Yeah,
and it's really
it's
worked.
kind of in
So
the western part of this. Yeah, yeah,
so
it's
there's a lot more
worked.
green hair streaks in the city now. Imagine having green hair streaks in your city. And the other thing he did was besides Mission Blues and so on, has been the Mission Street project with Western tiger swallowtails.
Oh yeah,
Realize they were
it's Market Street.
Market Street, I meant to say.
Yeah, yeah.
Like
They
the plane trees all along there that the Western tigers are happy on.
the
So he's promoted that and people's awareness of them. And that's been a cool project too.
Yeah, I know. I go to protest marches on Market Street, and whenever I see a tiger swallowtail flying by, like makes me feel good.
Yeah, you might want to get Liam for a program, Paulette. I think we'd all enjoy it.
Yeah,
his last name is O'Brien.
yeah.
Correct.
Yeah, I can lean on him too,
Okay. Yeah, I'll need his contact information and a little bit of information
Okay.
about it. But yeah. So,
Yeah.
uh, Vivian, do you know him as well, or?
All right. Yes, we've actually had him do a talk for us for the California Native Plant Society
Oh,
before.
okay. What did he
He
talk?
actually is considered the lepidopterist for our San Francisco chapter of the Native Plant Society.
hmm Yeah,
Oh, okay.
that's his county in the Lepidopterist Society.
Okay. Okay.
He's he's
Yeah.
followed on in the study of the butterflies of the city from a lineage of women who did that before him. There was a woman on the Long Beach Peninsula of Washington who discovered the Oregon silver spot in the state back in 19 teens, and her name was Agnes Vesey. There's a couple of subspecies named for her,
Mhm.
a checkerspot and others. But her daughter, Harriet Vesey, was brought into butterflies by Agnes. She went to San Francisco, became Harriet Reinhardt, and she became the doyen of butterfly people in the city, friend of all the old lepidopterists like Tilden and those people who saw the last Xerces blues. And she studied the history of San Francisco butterflies and wrote about it. Well, her um, apprentice was Barbara Deutsch, who became a great native plant butterfly person in the city and took Harriet's knowledge and built on that. And she became the great authority on San Francisco butterflies. And then she taught Liam. And they're all gone now, but Liam's there. And then his new book is all what he's learned from this whole heritage of studies of San Francisco butterflies.
Yeah.
Okay. I
Okay.
made note. All
Well,
right.
thank you everybody for sticking around for this. This is fun. I love talking butterflies.
We tend to do that.
Yeah.
So any other questions, comments? Okay, well, I just got a, message in the chat from Vivian with, I think so, with, yes, with
I was
Liam's.
just giving you Liam's email.
Yeah, so I'm going to take a note of that. Thank you so much, Vivian. And Stuart, thank you so much for this very, very interesting, exciting talk where it's really thrilling to see the work that you're doing. And we look forward to following it in the future.
Yeah. We're looking forward to seeing what shows up this
Yeah,
spring.
we may
Just
have
a
to.
phenomenal talk, Stuart. And thank you so much for bringing an old
Yeah.
guy's dream into reality.
Okay. Well, it's inspiring. I'm also inspired. I'm also inspired not just by your butterfly wanderings, but your beer wanderings.
There
I'm
you go.
afraid it's all gone. No matter what you do,
It's okay.
pint runs out.
Like in your big yearbook, it was like, okay, he's going after this butterfly, but what beer is he going to find there?
Well, you may if you're going to meet in the Presidio area in the spring, maybe make it more widely known, and you could get some volunteers coming down and checking
Well,
it out.
That's a good point.
okay,
Right.
Something to think about.
okay.
Got a
Yeah,
good point.
we'd love the opportunity.
Let's
Mhm.
take a field trip down. But I have to warn you, I go by Amtrak.
That's
Oh,
we can yeah,
So
we can
that's
work
fun.
that out.
let's fill up a train car and take a bunch down there.
Oh,
Get in Stewart's way.
the Butterfly Express.
There you go.
Okay,
I
well,
like it.
Okay.
next month we're going to have our holiday party, and so it'll be a chance to socialize. And all of you are welcome. Vivian, Stewart, anybody else who's Not a member now. Doesn't matter. We'd love to see you come January. So stay in touch, look at our website, and we'd love to see you. So, okay, thank you so much again, Stuart. Wonderful talk. And it'll be on our website, the recording as well. So good night, everybody.
Bye, everybody. Thanks.
Nice to see your face, Vivian.
Yeah.
Thank you, Stuart.
Ciao.
Good night.
Bye.
Adios.